So the characteristic feature of the metaphase, you can tell that is the metaphase. Is this the line? The chromosomes are in line. The homologous chromosomes are in line, right? So this line of the chromosomes are called the metaphase plate. And also note that the astral microtubules attach to the cell membrane. So these are the astral microtubules or astral microtubules. And also in the earlier phase, the microtubules or the kinetochore microtubules mainly attached to this middle or the kinetochore, and then these two sister chromatids are separated because of those kinetochore microtubules. Those mainly pull the chromosomes apart to two sister chromatids apart. So that is it mainly in the metaphase. The homologous chromosomes are in line at the metaphase plate, and in this phase the checkpoints mainly check for the kinetochore. Those mainly needs to be firmly attached. The kinetochore needs to firmly attached to the centromere. Sorry, not centromere. The spindles mainly needs to attach to the kinetochore firmly. So if that is not firm, if there is any problem in those spindle binding to this kinetochore, then the chromosome will not be separated properly, and this will result in anomalies. For example, non-disjunction. So we will be seeing the abnormalities in the mitotic process. So the non-disjunction will happen if there is any problem in the segregation of chromosomes. So moving to the next slide. So in the anaphase, the sister chromatids mainly separate. As a result of this separation, we get two sets of the genome, or the two sets of the same chromosome that are duplicated, and we get two diploid daughter cells. So just know that if that is the sister chromatids are attached by a protein complex. So coenzymes are mainly protein complex. Those mainly holds the sister chromatids together. And in this phase, there's another enzyme, separase. From the name, you can guess that separase mainly separates the sister chromatids. The separases are the enzymes that falls under the cysteine protease enzymes. So those are mainly have very different and important function, the cysteine protease. But just know that separase mainly cleaves the cohesion proteins so that the chromatids can segregate. So in this anaphase, the sister chromatids are separated in half, and also in the anaphase the cell elongates to actually produce two cells. We'll see that. So in this figure we see that the chromosomes are separated. So how those are separated? Those are mainly separated by motor proteins. So motor proteins are the kinesins and dyneins. Those are mainly main two motor proteins. Those mainly reels the kinetochores. That means those motor proteins attach to the kinetochore, and they spools those two uh, chromosomes after the sister chromatids are separated by separase enzyme. And also you can see these microtubules are disassembling because they are not needed now because chromosomes are separated, and cell also elongates in at the end of the anaphase. So this is the anaphase, separation of the chromosome, and how those are separated. The sister chromatids are separated by separase, and the motor protein mainly reels or pulls those kinetochore at the two sides of the cell, and the microtubules are disassembled because they are no longer needed, and cell mainly elongates. in this at the end of the anaphase. So at the next phase, which is the telophase, the main characteristic is this portion. This mid in the middle, there's the cleavage furrow. That means the that means there's this. If you can see this structure, that are mainly the microfilaments, if I'm not wrong. So those microfilaments mainly, and the part of the cytoskeleton mainly uh, do the function of the cytokinesis or telophase, pinching the cell into two half. So this in the cleavage furrow, that means the filaments are pinching the cells into half and producing two individual cells. And also, if you can notice that the nucleus are again visible, that means the nucleus reforms again to store the separated chromosomes. So at the telophase, at the end of the telophase, the mitosis is complete with two genetically identical nuclei. So cytokinesis is mainly the separation of the cytoplasm and the plasma membrane. It is not a mitotic phase. As I mentioned earlier, the mitosis is not part of the cell division, but it is necessary to know that how the cell mainly divides into two and separates into two, how the cytoplasm is separated. So uh, that's mainly the main part of the telophase. The nucleus mainly reforms again, and you can see this mitotic mainly spindle mainly are not that visible. That means they're disassembled, and there is one centrosome, two centrioles at each side of the cell, and also the formation of the cleavage furrow. That means pinching, cutting off the cell in the middle. So those are mainly the characteristic feature of the telophase. And cytokinesis is that at the end the the cytoplasm is separated. So that is in a nutshell the 5 stages of the mitosis, resulting in two identical daughter cells.